covered by this bar here. But anyways, so this is some of the oldest known eukaryotic fossils. They're pretty small. This is a 50 micron scale bar, so this entire thing, maybe 150 micron, represent a single cell this is from more than 20, 30, almost 30 years ago, reported from rocks about 1.7, 1.8 billion years old. That is interpreted as a single cell. This is a recent paper by Emmanuel Javot in Belgium. And this is a SEM picture of the same group of fossil showing the wall, the sculpture on the wall. This is a single cell organism, but the cell wall has the type of structure, the spines, for example, and this reticulate pattern that requires the machinery of eukaryotes, and particularly actin-based cytoskeleton, to manipulate the cell membrane to make those spines and to make this type of cell wall. So they're interpreted as eukaryotic fossils. Nothing like this is found in older rocks. Okay, so we take this as a minimum estimate of the split between eukaryotes and their living systems. Those fossils might be stem group eukaryotes, we don't know, or could be chrome. In the Tonian, so this is the other end of the Proterozoic, we have examples of this macroscopic, so the millimeters, several millimeters, sometimes centimeter in size. The 4 examples are all coming from the Tonian. They're macroscopic, like I said. Some of them are just a little sausage-like structure, but others just have simple differentiation, meaning structural differentiation into different structure. For example, they hold fast. Some of them are branched. Some of them have little balloon-shaped structure, and then a sort of kettle or a stipe that hold the balloon and are attached to the sea floor. They're uncertain in terms of phylogenetic position within the eukaryotes, but I think most of us agree that they are eukaryotes. It's just we don't know where in the eukaryotic tree to place them. Whether they are animals, there's a debate whether any of those could be animals, or algae. If they're algae, what kind of algae, right? Green, brown, etc. But there are a handful of Proterozoic fossils, all going back, most of them going back to about 1 billion years ago, at the end of the Borean billion, that can be confidently placed in the phylogenetic tree, at least in large scale. Here's one example that was published by Herman, 1990, a long time ago, and Nick Butterfield, 2004. What they're arguing is that this filamental structure, it is a tubular structure. At one end of the tube, you begin to have some transverse subdivision that is interpreted as reproductive structure. So a tubular structure, when they're ready to produce spores, they kind of separate part of the tubular cell into a compartment where they produce spores, and usually at the end of the filament or at the end of the tube. And sometimes they divide the tubes into large and small segments. Both of these structures are comparable to a living group of algae called xanthophyte, which belong to the group of stromatophytes that I mentioned earlier. So indicating that stromatophytes and the xanthophyte, or common name of this is yellow-green algae, at least going back to 1 billion years ago. Here's another example that we published just a few years ago. This beautiful preservation. This is a 100 micrometer scale bar, 0.1 millimeter. It shows the type of branching. So this is repeatedly branching. This is a branching thalli, or algothalli. This is one of the specimens that we were able to extract from the rock. These are preserved in mudstone. We just dissolve the mudstone with hydrofluoric acid. We can remove the branch, and the type of preservation at very close to each branch, there's a cell division. For example, here you can barely see it, and right here. This type of branching reminds us of a cladophoran green alga. Does the same thing in living green algae. So it's interpreted as, and actually named after cladophoran Protocladus, about 1 billion years ago, interpreted as a green alga. So we have the yellow green, we have the green, we also have red alga going back to 1 billion years ago. This again is a piece of work by Nick Butterfield. More than 25 years ago, he reported those structural from rocks about 1 billion years old from Canada. The type of cell arrangement, the paired cells, and the swollen reproductive structure at the end of the branch interpreted as a structure that is comparable or similar to some living red alga known as Bangiophyte. So this is named after modern Bangiophyte, Bangiomorpha. Number 4, so I've talked about yellow-green, a green and a yellow algae. A fungi also has fossil record go back to 1 billion years ago. This is a recent study by Lauren et al. So here is a fossil with a spherical structure that's compressed, that is attached to some filamental structure. This spherical structure is interpreted as a spore, and the filamental structure interpreted as a hyphae, a sponge. In fungi, this is some living fungi for comparison. So this is a hypha, or the filamental structure. This is spore. This is 70 micron. Forgot to put a scale bar here. This is about 100 micron in size. So similar in size, and it's interpreted as a fungus. Okay, this is a little younger, Tonian in age, maybe 750 million years old. This little thing known as vase-shaped microfossil. It has one opening on the top, and a structure look like a vase that you put your flower and other things in it. What interesting about this vase-shaped microfossils, many of them have little holes on it. Sometimes sort of half moon shaped, sometimes circular shaped, and those fossils are interpreted as amoebozoans. Amoeba sometimes make this test, and they can be organic, they can be agglutinated, they can be just collecting particles from the environment to make this test, and this is interpreted as an organic test that actually was these holes are drill holes made by predators. So it's presumably some eukaryotic predators preying on these eukaryotes, a process known to biologists as eukaryovory. So this is the evidence not only for the organisms, but also for the ecological interactions among organisms. So, putting the fossils in the phylogenetic tree that is based on living organisms. This is a tree of eukaryotes. So I introduced 5 of them, all pre-cryogenia. So this is a pre-glacial divide. If any of this interpretation was correct, then we have to accept, based on the topology of the phylogenetic tree, that the major branch of the eukaryotes, even for those